welcome to this meeting of the Royal Statistical Society's Professional Statisticians Forum. My name is Trevor Lewis and I will be chairing today's meeting. I'm a member of the Professional Affairs Committee of the RSS, which is the committee at the RSS responsible for professional matters and as such is responsible for the Professional Statisticians Forum series of events. One of the themes of the PSF is to present how statisticians, using novel methodology, can impact on the quality of work undertaken within their organisation and beyond, and today's presentation fits nicely into this theme. Our presenter today is Michael O'Kelly, who's the Senior Statistical Director with IQVIA, Advisory Services Analytics, who will be talking on missing data in clinical trials, making the best of what we haven't got. Before handing over to Michael, let me cover a few administrative points. Michael's slides for today's meeting will be available from the Professional Statisticians Forum webpage. So that's www.rss.org.uk/PSF. Following today's meeting, and also following today's meeting, there will be a webcast placed on the past events subpage of the PSF webpage. A brief comment on the structure of today's meeting. Michael will give his presentation and then there will be an opportunity for questions and discussion. However, Michael has indicated he's happy for you to raise questions for clarification during the presentation, but maybe if your question might require a longer discussion, maybe you'd want to hold it until after the presentation is complete. So all participants are initially muted. When we come to the stage where you might want to ask a question, then you can unmute to make your contribution, but please, having made your contribution, if you would remute, it would be appreciated. If you would rather ask your question in writing rather than verbally, then alternatively, you can ask your question by entering it into the conversation box, which is on the bottom left of your screen. If the conversation part of the screen it doesn't appear straight away, you'll see an icon on the bottom left corner of your screen looking like a message symbol. If you click on that, it will open the conversation box and you can type your message, your question in there. Maybe most questions will be raised at the end of the presentation rather than during the presentation. Okay. So let's make a start. I'm pleased to introduce today's presenter, Michael O'Kelly. Michael is a member of the RSS and also of PSI, Statisticians in the Pharmaceutical Industry. He coordinates a special interest group consisting of academics and pharmaceutical industry statisticians that shares training materials and software for working with missing data. With Bodana Ratic, he has published a book on missing data in clinical trials, which is published by Wiley. Along with others, he was also an author of a proposed best practice for projects involving modelling and simulation, which was adopted by PSI in 2017. In addition, last year, Michael received the RSS PSI Award for excellence in pharmaceutical statistics for his work in both missing data and best practice. So it's with great pleasure that I hand over to Michael to give his presentation entitled Missing Data in Clinical Trials, Making the Best of What We Haven't Got. Over to you, Michael.
Thank you very much, Trevor. And thanks to yourselves and the organisers and RSS for this opportunity to share my thoughts. So the the main thing I want to do in this talk is to convey an idea, as I see it, of how ideas about missing data in clinical trials have developed over the last dozen or so years, with a bit about the background before that. And then, having hopefully done that, I'll cover a number of topics of special interest with regard to missing data in clinical trials. This being a professionals forum, I've included a little bit about what we've done within my own company to try to improve practice with regard to missing data. Because I am a member of this group that shares software, and in the hopes of improving methodologies for missing data, I'll have a word about resources now available to you, particularly resources which our group has made available. And then 3 other selected topics, assuming that I have time at the end, I'd like to mention briefly. That is, one is prevention of missing data. Particularly enthusiastic about trying to use evidence from historical data when planning for future trials that may have missing data. And then finally, a note to encourage bearing in mind interpretability when planning for clinical trials with missing data. I'd like to acknowledge a lot of people who helped to contribute to the book co-authored with Bogdana Ratic, including Sarah Hughes, Belinda Hernandez, Ilya Lipkovich, also James Roger, who's authored or co-authored a lot of extremely useful software to do with missing data, which he has made available. And the group mentioned by Trevor, the Drug Information Association Scientific Working Group, which shares that software at that website you see there, and Gary Cook, co-founder of my company Quintiles, now IQVIA, who's given us regular advice. He's at the University of North Carolina. Okay, so to set the scene even before the dozen years that I'm going to cover, early work on missing data was done in the context of surveys and censuses for the most part, as you can see from that quote from Donald Rubin back in 1977. And it may, certain emphases in the way people have been thinking about missing data could be due to that, perhaps. So that you might argue that for surveys and censuses, it may be possible to argue that missingness, the fact that data is missing, is independent of the unobserved data, given the data that are observed. And that's really, in a nutshell, the definition of when data is missing at random. If we feel we have enough information in the data that are observed to in layperson's terms, to tell us about, to allow us to model the unobserved data, then if we can make that assumption, then we can call our data missing at random, and that's due to Rubin and colleagues. But in clinical trials, on the other hand, there is an intervention, which wouldn't usually be the case with censuses and surveys. There is an intervention, but there are other considerations that kind of make clinical trials different in character from censuses and surveys. Public safety is at stake. Clinical trials are usually hypothesis-driven rather than exploratory. They will often attempt to make some kind of causal inference, and there is an urgent need to control the rate of false positive findings. And all those factors kind of conspire to make the missing at random assumption somewhat more difficult to go with in a clinical trial. Stakeholders, as they say these days, stakeholders may assume that data after someone withdraws from a clinical trial must be worse in some unknown way than data while the subject was in the study. And that amounts to saying that we can't really assume that their outcomes would be missing at random after they leave the study. There's a good argument that they could be missing not at random, as it's called, MNAR. All this is not to say that Rubin didn't find examples of missing not at random among the surveys with which he started his work. He has at least one nice example that I came across back in 1976. But I think it may have affected the emphasis in people's thought about missing data in clinical trials. And let's go with that assumption of missing at random for the moment, so we can understand it a bit more. An important corollary of the assumption of missing at random is one that's maybe not exactly immediately obvious or intuitive. The corollary is if, given the observed data, the probability of an outcome being unobserved is independent of the value of the unobserved outcome. Now that's the formal definition of missing at random. If this is so, then where the background data is similar, so the unobserved outcome cannot be systematically distinguished from the observed outcomes. An implication of that, then, is that we should be able to model the unobserved outcome using observed outcomes as a basis. And that's a corollary that makes missing at random very attractive if we can assume it, because it allows us to do all kinds of things helpful things in terms of giving everyone an idea of what the missing data was like. Okay, so to move to 2007 roughly and onwards, at that time, the International Conference on Harmonization, ICH, had already issued, some years ago, the guidelines, Statistical Principles for Clinical Trials, and that had a lot of valuable points in amongst its text with regard to missing data. It urged the prevention of missing data. It noted that there are no universally applicable methods for handling missing data, and that doesn't just mean you have to look at your missing data and figure out which particular method will suit it. No, what they were saying there was that you cannot cure missing data by any particular analysis. It's always going to leave an element of uncertainty in your results. They recommend investigating the sensitivity of results to the assumptions of the method handling it. They suggest that the methods of dealing with missing data should be predefined, and they note the importance of making use of reasons for withdrawal, as stated by subjects in a clinical trial. So why is missing data such a big issue? Well, very simply, often it's the last visit in the clinical trial that's the one at which clinical benefit is credibly measured. And for that, we have missing data if a subject drops out of the trial before that last visit. And the practice for missing data in the first decade of this century would have been fairly, let's say, unadventurous, basic. Often the last observed value was used, known as last observation carried forward, or LOCF, or perhaps some poor value such as maybe baseline might be carried forward, baseline observation carried forward, or BOCF. For multi-part scores, perhaps a representative value was planned to be used if parts of the score were missing, and that's often still used. However, there was a growing use of more sophisticated methods which did make nice use of the missing at random assumption, in particular the use of mixed models in the repeated measures setting, known as MM or M for short, was growing. there was also a growing understanding that standard multiple imputation gives the same results as MM or M, essentially, in that multiple imputation, or MI, and MM or M model from the observed data and thus assume missing at random. So, given that, I'd like to paint a history of what happened, as I see it, between around that time up to right now. So, the general attitude was that missing outcomes had to be handled in some way, and hopefully without disturbing the general running of the trial and the analysis of results, and was often left up to the statistician. Then, with the coming of guidance in 2010, and with the growing of ideas about missing data, everyone began to realize that a fuller account should be taken of missing outcomes, taking into account the fact that after all, people who left a study would often have stopped the study treatment, meaning some inherent and maybe immediately unmodelable change in outcome, certainly unmodelable without some assumptions. Finally, in the last 6 or 7 years, a further development in thought came about, where it didn't start in 2010, 2011, but it became more generally realized that how you take account of missing data should reflect what you want to estimate in the study. That the assumptions you might wish to make or feel you should make about missing data may well change, be dependent upon, and affect what you estimate at the end of the study. So, just to recap, this missing data at the beginning regarded as something to be handled, and a variety of methods, such as we just mentioned, were employed. To help talk about the next stage, I'm just going to talk about the use of MM or M and MI, that's mixed models of repeated measures and multiple imputation, a little bit. And to help me do that, I'm going to use a sketch plot which originates with James Roger, who I mentioned at the beginning of the talk. And I find this very useful to understand the implications of missing at random as implemented in these popular frameworks. So here the vertical axis is a response where lower is better. The horizontal axis is time in the form of visits in a clinical trial. The blue line represents or follows mean values for the control group, which is placebo here. And then the green line, we can see it, shows the means for visit J for treatment J and visit K, with the lines being dotted for the sketch plot here, where our sample subject has dropped out of the study. So the subject is observed for visits 1 and 2, but after that the subject has left the study, and our task is to model their missing data. So there are a number of things that this sketch plot helps me to understand about the inference you get when you assume missing at random in the frameworks of MM or M and MI. First of all, as is very often the case, our sample subject here at visits 1 and 2 has values that are somewhat worse than the mean for his or her treatment group. You can see that the sample values are indicated by stars, and there's a dashed red brown line between the star and the mean for the treatment group, indicating a kind of residual for that subject. And because under missing at random, it's implied that you use all the data you have observed in modeling the missing data. Fitting with this, you'll see that the both the MMRM and MI then infer a similar relationship between that subject's mean for their imputations, because we're looking at imputations here, and their observed data. So you can see at visit 3, the mean for the imputed values for that subject is somewhat worse than the mean for that subject's treatment group, and that continues for visits 4 and 5. But you will also see that as time goes on, the mean for the subject's imputations under this in this sketch plot drifts toward the mean of that subject's own treatment group. And that's because under MI and MMRM, a subject's history of outcomes influence or have an association with the imputations via the between subject correlations, or within subject correlations between visits, I should say. But those correlations between visits for a subject get weaker as time goes on, and hence the mean of the subject's treatment group tends to dominate the estimate of the mean for imputations. The second item that's of interest to me here is that you'll see that the distribution of the imputations, which is represented by variance on the normal distribution portrayed sideways in red, the variance of those distributions gets larger as time goes on, and that reflects the fact that it's almost always in a clinical trial the variance of outcomes does increase over time from baseline, but also the weaker correlations would contribute to that increasing variance. Okay, now that's all very well. You could say that in some ways it takes account of the missing data, because the estimate of efficacy, say this is an efficacy score for this subject who dropped out, does take account of the fact that that subject was worse than average for their treatment group when they were observed, and makes the imputed values for that subject similarly worse. However, as we mentioned earlier, the subject is, after all, no longer on randomized treatment. Why should we model missing outcomes based on that person's own treatment group? How realistic is that? And there are many who would say that they would be interested in such an estimate, the estimate as if subjects kept to their own treatment group, for instance. But for many, to take full account of missing outcomes, it would make more sense to impute clinically plausible trajectories post-withdrawal. So there sprang up a number of methodologies to allow us to do that in quite a, I would see as quite a clinically plausible way. So one of the ways of doing this would be, this is the same plot again, but now we could instead of modeling subject outcomes post-withdrawal as being based on their own treatment group, you could model them as if they were a member of the control group, and in that case their outcome would look like a rather better than usual outcome to begin with after their withdrawal, but would, as we saw for the corresponding to the other plot, would tend to drift towards the mean for the control group over time, and that could mirror quite a clinically plausible outcome. And indeed, a lot of the work in the group I'm involved with was devoted to making software for such models available so that they could be easily implemented for a variety of endpoints. Okay, but I want to cover a kind of a further development, as I would see it then, in thinking about clinical trials and missing data that has happened maybe since about 2000, although there was some important papers written on the subject before that. So this is how you take account of missing data should reflect what you want to estimate. So what do I mean by this, or what is meant by this? Well, the kind of change in thinking could be seen by example, perhaps. If we take the following statement, one that I would have made myself not many years ago, a completers-only analysis is biased because completers are likely to be healthier than those who withdrew from the study. And you can see, for instance, that, say, a new treatment that had some nasty side effects could look good if you used completers only, because maybe very hardy subjects were able to keep on the study despite those side effects, and the new treatment might look extremely good and might not suffer from, in its estimate of efficacy, might not suffer from its side effects. They might not attenuate that estimate. So what is wrong with that statement, if anything? What people began to realize is that it lacks a precision. And they asked, well, is a completers-only analysis biased if indeed what's planned to be estimated is the effect on those who could take a full course of treatment? So people began to realize that you cannot throw around the term biased unless you quite precisely define your terms. Biased for what? Biased for what estimate? And so it's now generally realized that there is a fairly close relationship between missing data and what you want to estimate. The message is that questions like, is there missing data? Is data usable? Or perhaps I should be saying, are data missing? Are data usable? Should my approach to missing data, or does my approach to missing data lead to bias? They all depend on what you want to estimate. For which a nice term from Latin grammar is the estimand. Other questions also depend upon the estimand, like, should we collect data after a subject withdraws from treatment? It could be that what we want to estimate has, as it were, some interest in that also. What assumptions should we make about missing data? And those what assumptions are useful and what aren't could depend upon what you want to estimate. Also, what sensitivity analyses should we have? If we have a very precise idea of what we want to estimate, and we can estimate it using the data we have, maybe we don't need a sensitivity analysis. And what's the best study design is going to be influenced by what you want to estimate too. These are kind of self-evident truths, if you like, but they were truths that weren't really generally recognized even just a dozen years ago. Some of them. Anyway. So what estimate should we have? And that, of course, then depends upon the study objective. That, again, may sound a bit trite, but this whole discussion, one positive side of it is that statisticians and the people who use the results from clinical trials, often the clinicians, clinical experts, and patients, are kind of forced to get into closer dialogue. Another consequence is that people who work in the area of missing data are beginning to think that the word missing is too small a word for what we used to call missing data now. Why is that? Well, can you think of an example where data is actually available, but it's unusable for the estimand? And you probably can think of one, but here's one. For instance, take the situation where most subjects on the control arm took rescue, while few on the experimental arm did. If that were the case, then you might want to estimate in the trial the treatment effect were no rescue taken, or in some way ignoring the effects of rescue. So in that case, you might have collected data and data available for subjects who, during the periods in which they took rescue, but that data might be unusable for your estimand. So it's not exactly missing, but it's at the same time it's difficult to find a way to use it. Okay, that was a sketch of how I see views on missing data have changed over the last dozen years. But as to give a few words on what we have done in our own organization to improve practice for missing data. First thing I should mention is that the company supported the authoring of a book on missing data with my colleague Bogdana Ratic, which we use in the company and is used by other companies also. On the less fancy side, we do a lot of training. We have 8 modules on missing data, each at least an hour in length. It may sound over the top, but in fact we didn't have difficulty getting buy-in to provide that training and for people to attend it, because missing data is a problem that really, or an issue that happens for every single clinical trial with which we're involved. We've also given practical workshops where statisticians are given data which was derived from clinical data, and they have to implement various strategies to suit certain selection of estimands with using template code. And then people compare and discuss how outcomes differ depending upon the estimand and depending on scenarios for the subject who withdrew from treatment. We also, and this might be useful to other companies perhaps, we have a thing we call Missing Data Superusers. Because this is such a common problem, it was felt worthwhile having a so-called superuser in each physical office. That's what we aspire to. I don't think there's one in absolutely every office, but in most of our offices, who would be the first call for anyone in the office with a problem or question about missing data, with an experienced consultant on call to work with the superuser as needed. And in my experience, that would be about 60% of the time. And we find that this works quite well. You can make the business case because the issue affects most clinical trials, as I mentioned. But I wouldn't underestimate the work involved. The updating of superusers is something that's an ongoing requirement, and you always have new superusers, so new training requirements. But it is a huge efficiency saving to have spread the expertise about missing data throughout the company. Okay, that's just a note on what we've tried in our company with regard to missing data. A note then on some resources, and I'm particularly enthusiastic about this because of the work I do with that DIA scientific working group, as they're called, for missing data. First, should mention the MICE package in R, which is widely used, and then this the DIA group has, as far as I can see, an unprecedented collaboration and sharing of new software across industry and collaboration between industry and academia. Do have a look at the web page. It has lots of software freely available for download. Mostly R, but mostly SAS, but some R packages also. And I won't go through the list there, but it's lots of things which we find, I think, are used in practice quite a bit throughout the industry in clinical trials. There's also sample statistical analysis plans, which have been reviewed by the entire group, so that includes academics and industry statisticians, so it's both practical and theoretically sound, we would hope. There are training slides, training materials, and there's test data sets. So I'd urge you to have a look at that website and hope that it would help you in your practice to do with missing data. That's a picture there with our DIA working group page highlighted. Finally, I think in the resources section, I want to mention my favorite piece of writing on missing data. It's a monograph that was sponsored by or funded by the NHS by three of the great minds who are involved in missing data, Carpenter, Kenward, and Roger. I've given the link to it there, and it covers a lot in quite a short space, and it's full of good sense. I would recommend it highly as a very readable and rigorous piece of work. Now to my three kind of little topics at the end that I wanted to try to cover. First one is just about prevention of missing data, and forgive me if I don't go through all the list of things that you can do, but I would like, if possible, it would make life better for everyone if we as statisticians were aware, I would suggest, of the ways in which we can help to prevent missing data. Not just in trial design, of course there is that, but also trial conduct. And Sarah Hughes has done some nice work on that, and I think statistician supported conduct of clinical trials is becoming more common in terms of focusing on patient retention and prevention of missing data generally. But you can also choose sensible analysis strategies that take proper account of missing data. And I would also urge you to think about the future and consider collecting data after people have dropped out of or have stopped, say, refused treatment, if possible, if they can consent to being followed up despite stopping treatment. That can really improve the science in future trials in the area. Okay. The second topic of the three special topics I want to cover, this is takes a little longer, but I will try to cover it quite quickly. It's a slightly contrived example from insomnia, but there are other ones which are which I've come across where historical data can be really valuable in helping to plan for missing data or people who stop treatment at least. So in this case, we're positing a two-arm trial in insomnia versus placebo, where total sleep time is the efficacy measure, and obviously higher is better, and we're expecting a fairly substantial proportion of withdrawals. And for our purposes, we'll suppose that the experimental treatment has a mechanism similar to the ORAs, the orexin receptor antagonists. And it happens that in this case, but not just this case, in many cases, there is available some very apposite post-withdrawal total sleep time data for a similar treatment, which I was able to find in an FDA briefing document. And here is the plot, which is so useful, which I found so useful in this case. So this is a plot of a randomized withdrawal study. That's and the three lines are as follows. The black line with black squares are people who are on the ORA and were randomized to stay on it. The dashed line represents the means for subjects who were on randomized to placebo and to stay on placebo. And then the black line with white squares represents people who are randomized to placebo and randomized to the aura and randomized to switch to placebo in a blinded fashion at month 12. And you can see that while the trajectories do change at month 12 for all treatment groups, the trajectory for those who are randomized to withdraw from the experimental arm and switch to placebo changes more dramatically than the other two. And in fact, there is, you can see that the mean in the first visit after switching to placebo is actually worse than that on the placebo group. So you could, to try and explain how useful this could be, if you think in terms of how you might model outcomes for someone who withdrew, if you look at the trajectory that we see there, if we trace roughly the trajectory that we might get if we modeled withdrawals based on the control group, you can see it's sketched there in with the red line. Clearly, it's pretty clear that that is not, as it were, harsh enough on the as a trajectory for those who withdraw. There's still a gap between for the first visit or 2 or 3 visits after withdrawal between the typical subject who withdraws and the control group or the placebo group. Maybe some trajectory like this, a modified trajectory based on control, might work, but there's still a considerable gap there that maybe it's some kind of withdrawal effect, we don't know. This kind of plot could be really helpful in informing the kind of assumptions that would be realistic for those who withdrew. And it's perfectly possible to introduce some kind of a delta adjustment there at the first visit post-withdrawal to make the trajectory match more closely the trajectory we see here. So that just summarizes what I've just been describing there. So finally, just a word about interpretability. Assumptions about missing data will be most useful if they reflect reasonable scenarios and result in the clinically interpretable estimates. And that means that we're going to have to work very closely with subject matter experts, clinicians, and so on, to identify such reasonable scenarios. And although I have been talking about clinical trials here, I'm hoping that this could be useful to anyone who's attending this webinar who works in other areas. That thing about using historical data as evidence for your assumptions is, I would like to emphasize. And not only the intelligent reader will want a scientific justification for any missing data strategy, but if you're trying to get a drug approved, the regulator will need that too, and quite rightly so. And historic data can really help in justifying a strategy if it's reasonably used. It is kind of surprising how many methods for missing data produce an estimate that really is not of interest to clinicians, patients, or regulators. And it's a simple thing, but I would remind people the importance of asking, is what I'm estimating of interest, given the data I'm likely to collect? Finally, you'd be surprised how often historical data are available to help subject matter experts and statisticians identify likely outcomes dependent on events that interfere with the experiment. So to summarize then, I've tried to sketch out how thinking in practice has evolved over the years. I would recommend the use of some equivalent to the superusers that we've used in our own organization as a very efficient way of spreading the expertise for missing data throughout your company, assuming that it is that missing data is important in your practice. We have a role in prevention. You'd be surprised how much evidence there is from historical data to aid planning for missing or unusable data. And remember that missing data, where missing data are concerned, statistical methodologies should not be the dominant factor. What matters is that we get an estimate that is actually going to be meaningful to the readers of the report to which we have contributed. Okay, so I'd welcome questions from people at this stage.
Well, thanks very much, Michael, for that presentation. Very much appreciated. I just wonder if I could get the ball rolling by just looking at the first bullet on your summary slide, when you're talking about thinking and practice changing over the last 10 to 15 years. I wonder if you could comment on how the regulatory view has changed over that period in how missing data should be handled, and whether there is currently variation across agencies in how they would like missing data handled, and indeed within agencies where there's differences between therapeutic areas.
Thanks, nice question, and almost impossible to answer, except with probably yes, yes, and yes. But I think in practice, a big change that we have seen is the pretty unambiguous rejection of the assumption that we can model away missing data as a problem. And that in turn means that while people felt that they could assume we could model, we could assume missing at random, and we could therefore use a lovely model such as the mixed model for repeated measures approach, and that would solve our problems. Those have been explicitly rejected in papers by some regulators, and orally also by other regulators, and very prominent ones, and that would cover both EU regulators and US regulators. I'm not sure about Japan. So that's been a big practical change. But I think you could say that it's simply that it reflects that people are thinking more clearly about what we want to estimate in clinical trials, and that rejection of a simple, relatively simple missing at random approach follows that. Secondly, is there variation? I think the documents that we have, the official and semi-official documents that we have from the US and from EU, are reasonably consistent with each other. I would regard the EU missing data guidance as a very well written and clear document. It also has the virtue of being short. In the case of the US, we have a national research NRA, an FDA sponsored report whose points more or less coincide with those of the EU missing data guidance, with some very small differences. But that US document doesn't have the advantage of being official guidance, and in fact has been criticized by FDA itself in the last 8 years since it was published. So things in the US are a lot less clear because of that lack of an official guidance, but also, as you hint, the attitude and responses that one gets from the US regulator do vary surprisingly from therapeutic area to therapeutic area. And I think that seems to be the nature of the beast in terms of the FDA, you will even have certain regulators expressing views in papers that are which are not maintained when you get to individual judgments of new products, and of course that's to be expected, because all those papers are explicitly don't reflect the thinking of the FDA itself. So I think the other change that's happened over the last few years in regulatory thinking is one that's still ongoing, and that's the thoughts that are being developed in the writing of the addendum to ICH E9. That's the International Conference for Harmonization guidance that I mentioned at the beginning of the talk. That addendum is addressing that issue of what we want to estimate, and although a draft has been issued, it has received hundreds of comments, and it's quite likely that the working group that are involved in that, which includes regulators from EU, US, and Japan, and others, representatives of industry also, it's quite likely that the thinking in that draft will change before we get a final version of it. So there's a lot still up in the air about regulatory thinking.
Thank you, Michael. Thank you for that answer. I noticed there is in the conversation, there is a question from Mona. I don't know whether you want to just read that through. You maybe partially answered it already. I'll just read it out.
Oh, I see.
Thank you very much for the seminar. In your experience, what is the minimum percentage of missing data that would warrant using missing data imputation sensitivity analysis? Are there any guidelines?
Thanks for spotting that. Sorry, I missed that one because there was quite a few comments there. Yes, I think this is. I'm surprised that people ask this question, to be honest, because if it so happens that, say, you're testing some hypothesis in your experiment, and it so happens that the treatment effect that you're testing is such that, as it happens, your criterion for rejecting the hypothesis is almost reached or just reached, then even a very, very small amount of missing data is going to cast doubt on that. Now, this may not be very scientific, but that's the way certainly regulatory science operates, but it also where you have a borderline set of evidence, then the missing data is going to be a big thorn in your side, even if there's very little of it. So, as a practical piece of advice, I would say always assume that you need every bit of evidence you can get, and therefore always plan responsibly to include your missing data in the design of the experiment and its analyses. So, short answer is no, I don't think there is a minimum percentage. That, however, I don't want to be too blanket, because I've certainly heard examples where advice was given by some regulatory body, it may have been some time in the past, but that if there was only a very small percentage of missing data, this would not be a cause of concern. So on some kind of casual level, people may have received advice that they could ignore missing data for if it provided it was below a certain percentage. But in practical terms, I would think that's not great advice because of the situation I described there, where due to whatever cause, whether it's an attenuation of effect or simply because of the size of the trial versus the size of the treatment effect, you've got a borderline result, where even one or two bits of missing data could change the potential conclusion from your trial or experiment. Thanks, Mona, for that.
Okay. I just wonder, Michael, whether we should ask whether anybody wants to raise a question orally. We have a couple more in the conversation, but give somebody the chance to raise a question orally before we look at those. Okay. If there isn't, then maybe the question from, first of all, from Arthur Taylor. Can you please explain the role of the statistician in patient retention in clinical trials?
Thanks, Arthur. Yeah, I've listed a few of the particular ways in which statistician can affect patient retention more generally. Things like pointing out when enthusiastic designers of trials pile on hundreds of measures, and the fact that the patient burden may be such as to actually put people off staying in the trial. So that the statistician can bring that concern about patient retention to the general discussions about the design of the trial, quite apart from its analyses. Another way in which the statistician can help with patient retention. This is the subject on which I mentioned Sarah Hughes has written, and colleagues has written some nice papers. In Sarah Hughes's case and her team, she used past trials to estimate the subgroups more and less likely to drop out of a clinical trial. I think she used a complex of 3 different subgroups in an HIV study, and she was able to produce a heat map, not perhaps as she says herself, you'd like to have confidence intervals and so on, but she produced a heat map which the people monitoring the study and running the study found really useful in terms of helping them to focus on subjects who might be more likely to drop out of a trial and provide encouragement in any way they could to keep them in the trial. Obviously, there's 2 points to this, 2, if you like, limitations to what you can do. First of all, a statistician, or in fact, when running a trial, you cannot hope to eliminate withdrawals due to, say, side effects or lack of efficacy. It's the other kinds of withdrawals where monitoring strategies can hope to affect the rates of withdrawal. And the other thing is, it would not be useful or statistically principled to use a study of people who are likely to withdraw to then influence your recruitment strategy. Because if you recruit according to those who are likely unlikely to withdraw, you're recruiting a very special population, and your inference would be limited because of that. But there is still, I think, plenty of scope for statistical inputs to monitoring to help improve patient retention. And in the case of Sarah Hughes's work, they did reduce the withdrawal rate or improved retention dramatically in those who were withdrawing due to causes other than efficacy or side effects. So those are 2 areas then: design of the study and then actual statistical input to aid monitors. Maybe I should just mention, in addition to study design, our unique consciousness of how important missing data can be in terms of power and sensitivity of a study, can lend weight to other aspects of design that could improve retention, such as flexible consenting. I know I've certainly had that role sometimes, pointing out the benefits of flexible consenting in terms of maximizing the amount of data that we can collect for subjects who might not wish to keep on the treatment, for instance.
Okay, thank you, Michael. There's a further question on the conversation from Sue McKendrick. Would you recommend adding estimand approach to handle intercurrent events, missing data handling, sensitivity analyses into protocols we are currently writing, or should we wait for ICH E9 R1 addendum to be?
Yeah, I would have no hesitation in recommending that you add them now for two reasons anyway. One is that we've certainly seen practical reason, we've certainly seen regulators requesting the description of an estimand in protocols and statistical analysis plans that have been submitted for review to the regulator already. So the regulator themselves are not waiting for the addendum to come out. But the second reason is that as I've tried to explain, it just makes sense. If you plan a certain methodology for your missing data, then it should, or anyway, whether or not the addendum, whatever the addendum says, it should address what's wanted to be shown, to be estimated by the study, by the people who will read the study report in the end. So it actually should improve your study, its design, its analysis, and its usability, its usefulness at the end.
Thank you, Michael. Sue agrees, so that's good. Okay, are there any other questions that people would like to raise verbally, or are there any others to be added to the conversation? No, while we pause. Maybe I could just ask another question, Michael.
Mm-hmm.
You mentioned at the beginning of your presentation how considerations around missing data emerged in other contexts. I think it was in surveys you were mentioning.
Mm-hmm.
Presumably there is a continuing evolution of handling missing data in other sectors away from pharma and clinical trials. Is there continuing collaboration across these sectors of people working on missing data so that ideas and approaches that are being developed in other sectors are also being, if you like, incorporated in the way that people are thinking about missing data in clinical trials?
Now there's one that I feel inadequate to answer, actually, because while I've tried to be involved in sharing expertise within the pharmaceutical industry, I haven't been involved anyway so much in sharing across the disciplines. The one light I would see there is the work of Van Buren, I think is his name, who the author of the mice package in R, dealing with missing data, where the methods that can be implemented via that R package and similar ones are implementable in, say, the other pharmaceutical standard language SAS. Those methods are illustrated in his book by quite a wide variety of data, so that at least via that R package, users there's a common methodologies are being used across a number of disciplines. But you're right that, or at least it's an unfortunate truth that I don't think there is enough sharing across disciplines with regard to methodologies for missing data.
Okay, thank you, Mike. So it looks like we don't have any more questions. Just pausing to see. Oh, we do.
Yes, nice one. So I'll read this one out then, Trevor.
Okay.
Would you have any particular advice for statisticians operating in phase 2A or proof of concept versus phase 3? Well, I would, although when I have been involved in, say, consulting on design of phase 2A or proof of concept, my thoughts have sometimes fallen on deaf ears. I think people are, cost is an important aspect of earlier phases, and I think sometimes the idea of elaborate, and as they might be seen by the people involved, elaborate analyses of missing data at the phase 2A or proof of concept stage might be regarded as superfluous or de trop, as opposed to at phase 3. What I would say is that you can actually enhance the probability of success in phase 3 dramatically if you bear in mind the issue of missing or unusual outcomes in the earlier stages. As my second last subject mentioned there, if you can produce evidence to justify your strategy or the plausibility of your scenario, whichever, with regard to missing data in phase 3, that's gold dust. People are hungry for evidence that what you're doing isn't just plucked out of the air. And if you have evidence about what happens to subjects after they stop treatment from phase 2A or proof of concept, that is such a valuable piece of armor, as it were, for when you're designing your phase 3 and for justifying that phase 3. So I would say two things. First of all, for those earlier phases of development, this is a wonderful time for gathering evidence about what happens when people stop treatment, which can be used for your phase 3. So I would urge people to do that. It adds to the cost because you may be collecting data after the point of time where someone is actually taking the treatment. So it may seem a waste to those involved, but I would urge people to put the case for continuing to collect data after withdrawal. And the second thing is that you will have to have a decent estimate and accompanying strategies for missing data in your phase 3 study anyway, and it is really useful to have an idea of whether and how your estimate of efficacy will be attenuated due to that missing data in phase 3. And you won't have, there's no better place to estimate how efficacy would be attenuated due to missing data than in your earlier phases. So on those two grounds, preparation for phase 3 by evidence and preparation for strategy by seeing how missing data affect your estimate, those two grounds I would say are powerful ones for taking full account of missing data in earlier stages. Maybe I should add that sometimes the phase 2 results can be quite an important part of a submission, and they will always look and be more credible if they take full account of any missing data that occurred.
Thank you, Michael. We noticed that a couple of people had unmuted. We were hoping that might be that they wanted to ask a question. It seems not. And I could tempt another question by saying, looks like we've dealt with all of the questions. This time I think we have. So maybe I should bring the meeting to a close. Importantly, thanking Michael for an excellent and broad-ranging presentation and sharing his expertise in this area. Very much appreciated. And also thank all of those who asked questions, and also thank Michael for fielding a variety of questions based around his presentation. So thank you very much, Michael.
Thanks again for the opportunity.
Okay, thank you. Just to reiterate, the slides of Michael's presentation and a webcast of this meeting will be placed on the RSS website by the end of the week, and that is at www.rss.org.uk/PSF-past-events. So if you go to the PSF webpage at the bottom, you can click on past events and it'll take you to the webpage where you'll see the webcast and the slides. And maybe one final notice to people who attend these webinars, this series of webinars. The next PSF webinar will be next Wednesday on the 16th of May, entitled Excellence in Official Statistics, Engagement, Insight and Building Trust. And information about that webinar can again be found on the PSF webpage. So thank you all again for attending, and thank you, Michael, for your presentation. So we'll close the meeting there. Thank you very much indeed.